my honor today to introduce our seminar speaker, Dr. David Hein, Chairman of Pharmacology and Toxicology. Just going to hit a couple highlights of Dr. Hein's long and illustrious career. I didn't have enough time to go through all 100 and something pages of his CV, but suffice it to say, he got his PhD before I was born. But the interesting thing is that David Hein became chairman of a department at the age of 32 when he was still an assistant professor. So he was an assistant professor for 2 years and the chairman of a department, which I always find just absolutely amazing. That was at Morehouse University. In 1989, he moved to be the chairman in the University of North Dakota. And in 1997, he joined us here at the University of Louisville as the chair of pharmacology and toxicology here. Dr. Hein has contributed to over 260 publications, peer-reviewed publications, over 700 abstracts on his CV, and he's been on 54 PhD committees, 73 master's thesis committees. He's mentored 20 students and at least 12 postdocs. So those are the kinds of things that I really look for when I look at people's CVs, because I think training the next generation of scientists is really, really important. And one other thing I should say is that he's the director and co-director of the R25 program, which is one of the reasons we have so many people in the audience today. So with Dr. Kidd, obviously, is the other director. So without further ado, I'm going to go ahead and let Dr. Hein tell you about this topic.
Thank you very much, Dr. Beverly, and good afternoon to everyone, especially as Dr. Beverly said, to our R25 NCI Cancer Education Program students who have just begun the summer program, and this seminar series is a part of their training. And I appreciate what Dr. Beverly said about student training. That has been a passion of mine, and it takes up a lot of the positions that I currently have. So what I'm going to do today is tell a story. Dr. Beverly mentioned that I'm fairly old. And so we've been at this for a while, and we've gone in different directions. And recently we've gone into a direction looking at insulin resistance and metabolic disease. I'm not going to talk about that today. My colleague, Dr. Kyung Hong, presented information on that at our recent pharmacology and toxicology seminar, and my senior graduate student Kennedy Walls will be defending her PhD on that topic later this year. So, and this is a cancer seminar, so I'm going to focus on cancer. We've been interested in the N-acetyltransferases, both N-acetyltransferase 1 and 2. I'm going to focus this talk on N-acetyltransferase 2 so that everyone can leave the room in a reasonable amount of time. So I've been involved in pharmacogenetics for a long time. We have used the term pharmacogenetics as an umbrella term to include all chemicals that people are exposed to, including environmental chemicals. There are new terms now that have come about. Precision medicine is one of those terms, and now precision environmental health kind of fits perfectly what we do. We do laboratory studies in which we can translate and augment human epidemiological studies that often are hard to interpret. So we have been fortunate to collaborate with colleagues across the world in these studies, as well as increasingly colleagues here at U of L. So I'll show some of that as we go along. So I need to give a conflict of interest statement to begin. Our studies have been funded by a number of government, research foundation, industry, subcontracts with other universities, and internal grants here at the University of Louisville. They're all listed here. I'm going to present some topics that are related to these sources, but they don't conflict with what those agencies might have in terms of their commercial interests. Some of these were several years ago, and we're still using the money because of collecting closed-end studies. So basically, some of the studies that I'm going to show you today that are ongoing in the laboratory now are actually funded by many of these agencies. So as I, excuse me, as I mentioned before, human epidemiological studies that associate chemical exposures to cancer risk often are inconsistently validated across studies. And this becomes a problem because one study will say this is bad for you, and then another study will say, no, it's not bad for you. And it causes a lot of misconceptions in public health and in the community at large that there really isn't a solid answer. It's been our premise that the obviously that humans are genetically heterogeneous and they have different levels of exposure. So it shouldn't be necessarily an easy question to look at exposure and disease, but those studies can be informed and improved by taking into account the genetic composition of the individuals that are being exposed, and then how that affects the exposure, what specific chemicals are you being exposed to, at what levels. And so that's all gene environmental interactions, and that's the kind of studies that we do in the laboratory, and those are the kinds of studies that bring epidemiologists to us to participate in their large studies, which we have been fortunate to do. And so a classic example is cigarette smoking, especially in this state. You know, for many, I'm not that old, but for many years, lung cancer was, you know, not solidly associated with cigarette smoking. Well, that's been firmly established. Everybody knows that. But when we were starting our studies, no other cancers were strongly associated with cigarette smoking. We, along with our colleagues, I think, have firmly established that urinary bladder cancer now is strongly associated with cigarette smoking. Over the years, we made the bold statement that we thought breast cancer might be associated with cigarette smoking. We got summary statements back from NIH review panels that said, no, cigarette smoking is not associated with breast cancer. There's been lots of epidemiological studies that have shown no effect of cigarette smoking on breast cancer etiology or severity. So we argued that, well, if you look at the entire population of women, it may not be significantly observed. But if you segregate the women into genetic phenotypes that are appropriate for the carcinogens in cigarette smoke, then maybe you'll tease something out. And so we've come a long way with cigarette smoking and breast cancer. I'm going to give you kind of the latest update on that. And the other power of these studies is they can actually, if you see something, they can be evidence of causality. So a lot of times these epidemiological studies are association studies. Something is associated with cancer. You know, PlayStation use is associated with cancer. TV viewing is associated with cancer. These are all spurious associations. But if you can show causality, then that really means something. And so we've helped others to think that there is causality evidence from some of the studies we've done. So we translate our laboratory research to improve the design. We have also been very much involved in the design of these studies, and we have told them these are the kinds of markers that you need to assess in order to improve your study. And looking at these gene environmental interactions contributing to cancer etiology. Genes and environment, the combination is involved heavily in most disease processes. You need both the exposure and you need a genetic makeup that affects how that exposure causes the disease. So I'm going to talk about some aromatic amines and some heterocyclic amines. I've got some automation here to show you that I'm not too old. So we're talking about aromatic amines. That means aromatic groups with an amine attached, an exocyclic amine. And two very prominent ones that are present in cigarette smoke are 4-aminobiphenyl and beta-naphthylamine. And I'm going to show you some studies with these two aromatic amine carcinogens. Just an example of a very prominent occupational carcinogen is mocha, and I'm going to show you a few studies with that. And then there are a variety of heterocyclic amines. We abbreviate them. Here are three of them: FIP, IQ, and methyl IQX. These are formed. They are present in cigarette smoke, but they're more primarily formed when the proteins in meats are cooked at high temperatures, and the creatinine and other amino acids generate into these heterocyclic amines. And not all of the heterocyclic amines are the same. FIP has a pyridine structure. IQ has a imazole structure, and methyl IQX has is slightly different than that as well. So we look at all three of these. Okay, so I'm going to focus today on tobacco and diet as the sources of those chemicals I just showed you. But in studies that are ongoing in our laboratory right now, we're also looking at these compounds generated by fossil and motor fuels, colorants, and textiles. And this is a big area. Colorants, you may have seen in the popular press lately. Children's candy, other food products have a lot of these azo-based benzidine dyes in there that can break down to these aromatic amines. So we're beginning to study those. I'm not going to talk about that today. And then the reason that we've been funded by some of those companies is the fact that these aromatic amines are also present in hair dyes and in cosmetics. And I'm not going to talk about that today either. I'm going to focus more on tobacco and diet. But just to know, this is a big area for these aromatic and heterocyclic amines. And so just a bit of metabolism here. Here's the aromatic amine. This is just a generic one, an aromatic group with an exocyclic amine. And it's metabolically or chemically stable. It needs to be metabolically activated in an individual in order to produce a disease such as cancer. So one of the primary steps is the N-acetylation of that exocyclamine, catalyzed by N-acetyltransferases, and we're going to focus on NAT2, to produce the amide compound, which is non-toxic and is usually excreted. These aromatic amines can also be oxidized at that exocyclic amine by cytochrome P450s. The relative form depends on various different human tissues. CYP1A2 is prevalent primarily in the liver. And so you hydroxylate that amine and you get an N-hydroxyamine. And then interestingly, this then becomes a substrate for O-acetylation of that hydroxy group to produce an acetoxy. It's catalyzed by the same enzyme. This acetoxy is highly unstable. It's a good leaving group. It will spontaneously generate aryl nitrenium ion, highly electrophilic, that binds to nucleophiles such as DNA. And if that DNA is not repaired, it can lead to mutagenesis and possibly carcinogenesis. So we're interested in which aromatic amine is causing a particular problem. We're interested in the metabolism, and there is both a deactivation pathway here for N-acetylation and an O-acetylation pathway for activation. So we're interested in both. And then we're interested in measuring indices of these DNA adducts or mutagenesis or carcinogenesis. So one of the beauties of the field that we have chosen to work in, the N-acetyltransferase 2 gene, is that it's a very simple single coding exon of 870 base pairs. And from pharmacogenetics, many of you will know that There can be single nucleotide polymorphisms in genes. These are very common. They're the reasons for pharmacogenetic differences between individuals in drug response or drug toxicity. And so this is the 870 base pair. This is a cartoon, a linear cartoon of the 870 base pair NAT2 gene. And there are 10 prominent SNPs along this, this exon. And so you inherit from mom and dad a haplotype in which these SNPs are inherited together. So, for example, and these are two that are going to be very important that we're going to talk about. The NAT2 star 5B allele contains these three SNPs. And it is an allele that's very common in Europeans, European Americans. Usually over 50% of people have that haplotype. And then another haplotype NAT27B has this SNP 857A as well as the 282T, C282T. And this is much more common in Asians and in Asian Americans. So this is a crystal structure of the NAT2 enzyme, and you see these numbers here numbered from 1 to 10. Those are the sites of those SNPs, those 10 SNPs that I showed you on that cartoon. So what it tells you is that these SNPs are located all across the structure of the NAT2 protein. And most of them produce the same effect. But one that produces a different effect from what we have been working on is this one here at number 10. And we will show evidence of that in a few minutes. So we have been characterizing these various haplotypes and the individual SNPs in expression systems. We've used bacteria, we've used yeast, we've used COS cells, we've used CHO cells, and we've done it in human hepatocytes as well. So these are just some preliminary studies in bacteria. And the NAT24, that's the reference allele that has no SNPs. And then you'll see that this group of haplotypes, all of which have the 341 SNP, have very low activity. The 7 series, all of which have the 857 SNP, have lower than 4, but not as low as 5. So we're seeing genetic heterogeneity here in these alleles. So that gave us a hint of some of the things we're going to do later. So this is for N-acetyltransferase. It's with a pharmaceutical substrate, sulfamethazine, which is a anti-infective. And so I'm not going to talk about pharmacogenetics today either, but there's a number of drugs that are metabolized by this same system, cause differences in drug response, drug toxicity. We've been funded for a lot of those studies. I'm not going to talk about drugs today. I'm going to talk about carcinogens. And so here's an example of carcinogen, and it's the O-acetylation reaction. So this is the 4-aminobiphenyl after it's been N-hydroxylated by CYP1A2 to produce the N-hydroxyaminobiphenyl, and then the O-acetylation of that to the acetoxy to produce the DNA addict. And you see and these are the different haplotypes again, expressed in E. coli, and you see pretty much the same pattern for the O-acetylation. This is the reference, and the 5 is low, the 7 is lower than 4, but not as low as 5. Okay. So then we looked at the O-acetylation of some of these heterocyclic amines. And this is just an example of one of them. We looked at all of them, and I'm going to show you some of the other ones later. But just as a hint, looking at recombinant expression in E. coli, here's the 4, the reference, it's high. The 5s are very low. But the 7s, they're not only higher than the 5, they're as high as the 4, okay? So that was the first inkling that something's going on here with some substrates for this NAT27 series, which has that 857 SNP, that number 10 SNP on that on that figure. So on that figure, we've now closed in on the specific site of the amino acid change. So for the 5B, the one that has very low activity, this is the one that's present a lot in Europeans. It's a T341 SNP, which has an amino acid change from isoleucine to threonine at 114. It is associated with slow acetylator phenotype. It's common in Caucasian populations. And in studies that I don't have time to show you, we've shown enhanced protein aggregation and targeting for degradation with this particular SNP. And it's thought to reduce hydrophobic interactions between the beta barrel and the helix of the different domains of the protein. So that's the mechanism of that one. The NAT27B is different. It sits right here. And this is an active site loop that kind of flips into the active site. And the size of this amino acid residue can cause steric hindrance for the ability of this active site loop to shield substrates from coming in. So it does cause a amino acid change from glycine to glutamine. It historically with slow acetylator phenotype, but we've shown some substrate dependent activity. I gave you a hint of that with N-hydroxy-FIP, and I will give you a lot more information on that in a minute. Common in Asians, it alters the active site access and produces steric hindrance to some substrates, not all substrates, but some substrates. So we wanted to characterize these SNPs and characterize these haplotypes in various recombinant various expression systems. And I've already showed you some studies in E. coli. As I said, we've also done yeast, COS1 cells, CHO cells, and cryopreserved human hepatocytes. And so this is from study of the sulfamethazine N-acetyltransferase. Now we're looking at each individual SNP expressed in COS cells. So this is a mammalian cell. Here's the reference, and you'll see that the 341 is low. That's with the 5B, but the 857 and the 857 is also low. Okay, so this is a slow acetylator. This is a slow acetylator based on this prototype drug substrate, sulfamethazine. But if you look at the KMs are constant, and these are not statistically different from these, but this one is. The 857, again, is significantly different. It has a much lower apparent KM. And if we look at that SNP on O-acetylation, looking at KM, for some substrates, it doesn't affect the KM, but for other substrates like N-hydroxy-FIP, it does reduce the apparent KM. So there's something about FIP again, and there's something about this 857 SNP. And so this is further studies in COS cells. We're looking at the O-acetylation reaction for N-hydroxy aminobiphenyl. Again, this is the reference and all the different SNPs. Again, you see the 341 is low and the 857 is low. But if you go to N-hydroxy-FIP in the COS cells, just like we saw in the bacteria, the 341 is low. And now all of a sudden 857 is not low. It's as high as the reference. So again, all of these SNPs do not produce the same effect for all of the compounds. And if we look at, this is also in COS cells, if we look at the actual haplotypes expressed in COS cells, again, for O-acetylation of N-hydroxy-ABP, the 5B is lower than the reference. The 7B is lower than the reference. But if we look at N-hydroxy-FIP, 5B is again lower than the reference, but 7B is not. So this is now showing this in not only for the individual SNP, but for the entire haplotype and in additional expression system and a mammalian system, COS cells. So then we wanted to see, do these differences in genetic differences in the metabolism of these compounds, do they result in differences in toxicity, in particular genotoxicity, because we're looking at cancer. So we have DNA repair deficient CHO cells, Chinese hamster ovary cells. The reason we use that model is we want to have a strong signal of DNA damage for nucleotide excision repair. So these CHO cells are deficient in nucleotide excision repair. If we get any DNA damage, it's not going to be repaired. We're going to see it, at least by nucleotide excision repair. So it's been a very convenient model for us to use. We humanize the cells. We put in P450s, either 1A1 or 1A2, as well as the P450 reductase, and then we'll put in the reference allele and NA22 variant alleles like 5B and 7B. And we construct the cells. So we start with this DNA excision repair deficient cell line, and we put an FRT site into the cell line so that all of our transfections are in a single site on the genome. We then put in CYP1A2, and then once we have that line generated, then we can generate these the NAT24, the reference, and the NAT25B and the NAT27B. And we have this system we can look at differences in genotoxicity. So we do mutagenesis assays. The assays that we do are not that sophisticated. We're interested upstream. What is the effect of metabolism, genetic differences of metabolism on the toxicity? So we have fairly routine measures of genotoxicity that are used by laboratories across the world. One is a HPRT mutagenesis assay. HPRT catalyzes conversion of 6-thioguanine to a ribophosphorylated derivative that is toxic. It's located on the X chromosome, so the cell lines have a single copy. So if you have a mutation that occurs, the cell will be resistant to 6-thioguanine. So after you do the assay, you treat with 6-thioguanine. The cells that survive, they've been mutated. You simply count them. And then we've also measured these DNA adducts. We've measured them by multiple reaction monitoring in triple quadrupole mass spectrometry, LCMS. So you load the sample onto the LC, and this is an example. Here's one of those heterocyclic amines, methyl IQX, that has been adducted to the C8 of deoxyguanosine. When that gets knocked around in the mass spec, you will get a daughter ion. I guess you're not supposed to say daughter anymore. A child, I guess, ion that I'm old, sorry. That it loses the deoxyribose, and so on the ionization spectra, this is the adduct. It has a mass of this amount. When you knock off the deoxyribose, it has a mass of this amount. So we can identify with great precision this adduct in our cells after treatment. And then we can quantitate it very accurately because we spike the cells with a deuterated compound, deuterated adduct. It's a D3, so it has 3 more hydrogens. So it's 3 mass units higher. And when you knock off the deoxyribose, this child is 3 units lower too. So you know how much you spiked with, and so you can use this as an internal standard to not only identify, but very precisely quantitate the adducts that you're generating. And so this is just, and we have lots of data, but just to show you some of it, This is with methyl IQX, one of those heterocyclic amines. We're looking at mutants in the UB5 cell, the untransfected cell, and then the cell transfected with CYP1A2 only, and then a cell with CYP1A2 and the NAT2 reference, and CYP1A2 and the NAT2-5B. And we're looking at mutants as a function of methyl IQX dose. And you basically, this is negative unless you have the CYP1A2 NAT2-4, and then you get a very nice dose response. So this is for mutations. If you look at adducts, again, at 3 different concentrations of methyl IQX in the cell after incubation and measuring the adducts, there's really no adducts formed except if you have the CYP1A2 and NAT2-4, both at this dose 0.5 micromolar, this dose 1 micromolar, and this dose 1.5 micromolar. So it's a dose response and a gene response. That's very nice. And we see the same thing with IQ, basically the same data. Again, it's only the NAT2-4 that shows the mutations, and it's only the NAT2-4 that shows the DNA adduct levels, and as the dose increases, the adduct level increases, and it's only in the NAT2-4 where it's significant. So that's where we were up till a couple of years ago, maybe a year and a half. And then we decided that based on some of the studies that we were seeing with the 857 SNP and NAT2-7B haplotype, that we would like to see if that makes a difference in genotoxicity. So we've done that with a number of those chemicals I showed you now. First one was with beta-naphthylamine, and this is dissertation work from Mariam Habil in our lab, and was published late last year in December. And this is the data, and I just want, this is in vitro data, and this is in situ data. And there's a color code here. So CYP1A2 NAT2-4 is blue, CYP1A2 NAT2-5B is red, and CYP1A2 NAT2-7B is green. So if you look at the acetylation of beta-naphthylamine, you actually see that it's highest in the NAT2-7B at high concentrations. At lower concentrations, it's not as high as 4, but it's much higher than 5. So we're seeing that in situ, in cell culture, as well as in vitro. If you then look at mutations, and we did 4-aminobiphenyl, we had done a lot of studies with 4-aminobiphenyl in the past, and so we used it as a positive control here. So we're looking at beta-naphthylamine-induced mutants in these cell lines, and again, actually the highest mutations are in NAT2-7B, which commonly in studies is considered the same phenotype as 5B. So if you're looking at the role of NAT2 phenotype in cigarette smoke induced anything, and if beta-naphthylamine is a contributor to that toxicity, then if you just look at slow acetylators, you are mixing these and these, which confounds the data and often then biases the data to the null, that you don't see it. But if you understand this, then you can design the study and separate out the different haplotypes. And then maybe you'll see something with, in this case, particularly with the very slow 5B haplotype. So even more recently than that, this year, this is again part of Mariam's studies. We've looked at this occupational carcinogen, MoCA, 4'-methylene bis 2-chloroaniline. And again, this is looking at in vitro And the 7B is again higher than the 5. The same color scheme, I guess Miriam used. And if you look in situ, again, the 7B is higher than the 5 in situ acetylation. And then if you look, we've done a lot of studies looking at another measure of DNA of genotoxicity, which is double-strand DNA breaks, which is measured by a gamma H2AX immunochemical assay. You beginning to use much more routinely now. It's a measure of DNA damage, and here again, it's a dose-related response, and the highest is actually in the 7B. It's much higher than the 5, and if you mix these two in the same phenotype, it's going to confound your data. And this is mutations, and here the 7B is actually the highest for mutations, much higher than the 5B. So in even more recent studies now, looking at genotoxicity, well, looking at the O-acetylation and the genotoxicity of these N-hydroxylated heterocyclic amines, here's 3 of them: N-hydroxy-IQ, N-hydroxy-methyl-IQX, and N-hydroxy-FIP. And we're looking at the O-acetyltransferase activity. This is in yeast, actually. We're expressing the recombinant NAT24. In other words, there's no SNP, or a SNP with a gene with a single SNP, the T341C present in the 5 series, or the G857A SNP present in the 7 series. And the data is interesting here. So for IQ, the 7B is about the same as the 5B. For methyl IQX, the 7B is higher than the 5B, but it's still lower than the 4. And for N-hydroxy-FIP, again, we're getting to that FIP compound, the 7 is much higher, almost as high as the 4, higher than the 5. So I'm going to show this data 3 more times together with the genotoxicity. So this is IQ. We're looking at the genotoxicity, double-strand breaks with different doses of IQ in these cell lines: UV5 with CYP1A2, UV5, CYP1A2, and NAT24, 5B or 7B. So the UV5, there's no genotoxicity. With just 1A2 alone, there's no genotoxicity. With 5B, it's very low. That's not unexpected. With 7B, it's also low, which is consistent with this O-acetylation data. And it's really the 4 where you get the highest levels of genotoxicity. But if you look at N-hydroxymethyl-IQX, again, the UV5 and the CYP1A2 are doing nothing. Again, the 5 is low. But now the 7B is coming up there. It's much higher than the 5, but still lower than the 4. Okay. And then if we go one more, N-hydroxy-FIP. In this case, again, the UV5 is negative. The UV5 with 1A2 is negative. The 5B has some genotoxicity, but then the 4 and the 7 actually come together. So the 7, and actually it's significantly higher in the 7 than the 4. So it depends on the compound. It depends on the haplotype, and it depends on the compound. And this data really matches this data really nicely. So how can we use that data? Well, we were approached by the NCI because many of our initial studies were in urinary bladder cancer when we started. And they said they had this big study that was going to be carried out in Spain. And what was really great about this study is, well, it's unfortunate, the Spanish population at that time were heavy smokers. And they were using a tobacco product that was high in aromatic amine, a very dark black tobacco. So, and they were able to get really good exposure data, smoking data on this group. And then they wanted us to look at the NAT2 question because it still was not established at that time. There was no establishment between NAT2 and any cancer. But we worked with them on this study and we told them how it should be done, and so you don't misclassify genotypes. And this was an important enough study. It was published in the Lancet, and it showed for the first definitive time an increased risk for urinary bladder cancer in cigarette smokers who are NAT2 slow acetylators. And then we followed that up. That was a Spanish study. We followed that up with many of the same investigators in New England a few years later. And we saw the same thing in New England, although this time we added to it that we showed that it was a function of smoking intensity as well. So, and then we did a meta-analysis in this study as well, which is shown here. So this is a plot that plots odds ratios and 95% confidence intervals, looking at the effect of slow acetylator NAT2 genotype on different studies published in smokers. And those of you who know epidemiology, if the error bars cross the line, then it's not significant. So most of them are positive, and you can see the numbers here. But if the lower estimate is below 1, then it's not significant. So many of these were not significant, and that was the dogma at the time, that there's not a significant relationship between NAT2 phenotype and smoking and urinary bladder cancer in smokers. But this study that we did in Spain was very tight. And after that, then if you do the entire meta-analysis, including our data, then it became significant across all of these studies. In Europe, also in the USA, we did the New England study. So here was our very tight New England study, and that made almost significant in the USA. And then in Asia, it's also significant. And there's a lot more details given here. But basically, it was establishing, based on these studies, that yes, NAT2 phenotype modifies the ability of tobacco smoke to induce urinary bladder cancer, and it is a risk factor for urinary bladder cancer. And because slow acetylator phenotype is so common, it has a public health impact that's relatively high. So we got to that point, and then I briefly said this before, we wanted to shift to another cancer. And so some of those heterocyclic amines have been shown to produce breast tumors in rats, and actually aminobiphenyl has been shown to produce breast cancer in rats, mammary tumors in rats. So we said we would like to do some of the same studies that we did in urinary bladder cancer and shift it to breast cancer. And the reviews we got back, stick with urinary bladder cancer. Cigarette smoking does not induce breast cancer. If anything, it reduces it reduces weight, and weight gain is a factor in breast cancer. So if anything, cigarette smoking is actually protective. We don't want to tell anybody that, but it doesn't induce breast cancer. And so we made the argument. We said, well, based on some of the studies we're seeing, we think that if you look at a particular subset of the population, then it will show up. And so eventually we got funded. I think actually DOD funded it first. And actually this was a study that we actually carried out in collaboration with our colleagues in the Netherlands. They approached us and they had this data, and they said, we're not seeing it. We're not seeing a relationship between NAT2 phenotype and breast cancer in smokers. And so actually this person now Dr. Vanderhel, she came to our lab to conduct studies with us and to do genotyping in our lab. And we segregated out the very slowest acetylators, the 5B very slow acetylators from the other slow acetylators. And when we did that, we were looking at GST-M1 null as well, glutathione transferase. But we saw that the NAT2 genotype, especially the very slow NAT2 genotype, are increased risk of breast cancer. And so I just want to give you an example of how these studies kind of look like. And at this time, you know, the sample size is very small, but so you're looking at a function of increasing dose. For cigarette smoking, it's the number of cigarettes you smoke per day, or the duration, how long have you smoked? And so if it's a dose-related effect, you should see an increased risk. Well, if you look at this response in rapid acetylators, you see no positive trend. It's not positive. Even if you look in slow acetylators, you don't see a significant positive trend. But if you focus only on what we considered the very slowest acetylators, those 5B people, then there's a dose-related effect with a positive trend. And the same with duration. No matter how long you smoke in rapids, you don't see a positive trend. In slows, even in slows, you don't see a positive trend. It's getting close, not there yet. But if you focus on the very slow, then it becomes positive. So that's how we did that study. And then that stimulated a lot of other studies, a lot of other groups working. And so this was a meta-analysis study that was published. It again is looking at, it's looking at pack years, okay, as a measure of dose. And this is the relative risk. And if you're postmenopausal, well, actually either pre or postmenopausal, if you're slow acetylator, you have an increased risk of breast cancer if you're a smoker. And it's dose dependent, pack year dependent, whether you look at a meta-analysis or a pooled analysis. These are the slow acetylators. If you look at the rapid acetylators, it's not significant. So again, when all of these studies were done, they didn't segregate into rapid and slow. They just looked at everybody, and then that confounds the data, biases it to the null, and you don't see it. So then in collaboration with colleagues here at U of L and the School of Public Health, we wanted to replicate the Netherlands study in the United States. We looked at Kathy Baumgartner's cohort of non-Hispanic white women in New Mexico. And we saw the same thing. We saw smoking as a risk factor, especially in very slow NAT2 acetylator phenotype. So those confirmed those studies in a US population. So many of you are probably aware that for the issue of smoking, it's not just active smoking, it's also passive smoking. And how important is passive smoking? And trying to show an association between passive smoking and these cancers is very difficult. But, and this is not our study, but these are studies coming out of Canada where they have looked at side stream smoke or passive smoking in breast cancer risk. And based on our studies, which they cited, they found the highest risk in extremely slow acetylators. Okay, so that was the beginning of that. And then the Canadian equivalent of the CDC, they got into this and they did, this is an expert panel. And the Canadian expert panel on tobacco smoke and breast cancer risk. And they summarized the data. They looked at it as an expert panel. And the panel concluded that the association between active smoking and breast cancer is consistent with causality. That's based in part on our biological studies. and that the association with secondhand smoke and breast cancer among younger, primarily premenopausal women who have never smoked is also consistent with causality. So then that was taken up by the U.S. Surgeon General in the U.S., and they have basically agreed with those findings, and that's part of the U.S. Surgeon report as well. So it's getting more and more established, but if you look at, you just Google cigarette smoking and cancers, you may not see breast cancer show up, but it's there. So to conclude, I believe there's a strong consensus regarding a role for NAT2 in arylamine-induced urinary bladder cancer, but we're still working on studies to develop the role of NAT2 polymorphisms in breast cancer risk. And the inconsistencies observed that investigate these associations, and they're looking at NAT2 or smoking or breast cancer incidence. They may reflect differences in arylamine carcinogen exposure. Is it beta-naphthylamine? Is it 4-aminobiphenyl? Is it what is it that they're being exposed to? And then are they classifying the phenotypes and not just mixing them all up together? And these associations can, if they're modest, they can increase substantially when considered in junction with other susceptibility genes. We've done some of those studies actually with Vanderbilt University for other cancers, colorectal cancer, and NAT2 as part of a subset of a number of other genes really increases the risk for smoking and colorectal cancer, which is another one which is not real strong, but we're showing it. So these laboratory findings inform and strengthen human populations studies linking environmental exposures with cancer risk. So Dr. Beverly said I'm old, which is true. He also he likes students. I like students too. I've had many students, and they did all the studies. I didn't do studies. So our lab has been built with students and it's continuing to this day. We have 3 new students starting this summer. It's changing in the guard, but Adrian Fretland, a lot of the yeast studies, he actually came with me from North Dakota. This is from his wedding, where he married a local gal, a graduate student from Dr. Pro's lab, actually. And Matt Leff came with me from North Dakota. Yi Feng was my graduate student in North Dakota. He finished, and I gave him the project, and then he came here to the Cancer Center as a clinical oncology fellow. He had an MD and did his oncology fellowship here, and came back to our lab and did a number of other studies as well. This is an old picture. Mark Dahl he also came from North Dakota. Many of these studies Mark has done. And I'm not going to name all these students. Linda was very instrumental in the cell constructions, Janet in the 341. And some of these also worked heavily on NAT1, which I didn't tell you anything about. This is another picture. I mean, these students, looking at Janet and Kristen, I showed you some of their studies. They're like huge leaders in the pharmaceutical industry, make tons more money than I do these days. The 2 Jasons are also heavily in industry. Here's Mark again. Chris States has been a tremendous colleague working with us for many years, and we're very grateful to Chris. This is another study. You see Chris again. Postdocs, again, in the industry, have had wonderful medical students, Jeff and Ashley and Glenn, and then undergraduate students such as Susan here. And here we have Carmen, who is now, in the office of the president, Biden's cabinet. And Lori Milner, who's run a company here in Louisville. And then more recently, medical students, well, I forgot medical student there. Jeff Rice did great studies and Ashley as well. And these are all prominent physicians now. Jonathan Gardner, I think he's doing his residency here at U of L. Marcus Stepp, Angeliki, we did a Erasmus fellowship with her from Greece. Sam Carlisle, Cicely Allen, who's now doing her fellowship at Johns Hopkins. And of course, Mark always eats with us. He still eats with us. He's still there. And then I'd like, Dr. Kyung Hong has been working with us for the past 5 years or so, particularly looking at metabolic syndrome. Been an enormous contributor to our lab. Roel postdoc, who left for industrial position last year. James Wise, who just took a faculty position at LSU. Affy, who has won every award possible, and is probably going to go to Stanford for PhD studies. Miriam Habil, who did her PhD, is staying on for a postdoc. And then Kennedy Walls, who's our now our senior postdoc and doing a lot of the studies on metabolic disease with NAT2. So with that, I thank you for your attention.